So welcome to 22nd ICANX Youth Talk. So I am your moderator today for this webinar. So thanks so much for joining us. So I am Chen Ying Hu, an assistant professor from University of Wisconsin-Madison. So our topic today is the immunodelivery. So immunotherapy has been a paradigm shift therapeutic approach with dramatic treatment efficacy against various diseases in the clinic, such as cancer, autoimmune disease, and inflammation disease. So for the emerging immunotherapy, as all other therapeutic drugs, how to precisely and selectively deliver immunotherapeutics to the disease sites is always the key for the treatment outcomes. So today we have three outstanding rising star speakers to share their most recent research in this field. So our first speaker is Dr. Shijie Zhao from University of Washington, Seattle. And our second speaker is Dr. Feng from Indiana University, Bloomington. And our final speaker is Dr. Zongmin Zhao from University of Illinois, Chicago. And we also have great panelists to discuss the future of the immunodelivery. So our first panelist is Dr. Guojun Chen from McGill University. And we also have a graduate student trainee join us in the discussion, Ms. Sichen Yuan from University of Wisconsin-Madison. So, without further ado, I will introduce our very first speaker, Dr. Shijie Zhao from University of Washington, Seattle, and he will talk about targeted delivery of microbial metabolites for the immunomodulation. Brief introduction about Dr. Shijie Zhao. So he is currently an assistant professor in the Department of Pharmaceutics at the School of Pharmacy, University of Washington, Seattle. He earned his PhD degree in bioengineering from the University of Washington at 2018. From 2019 and 2023, he served as a postdoc fellow in the School of Molecular Engineering at the University of Chicago. He has authored over 30 papers, including publishing in the high-impact journals such as Nature Biomedical Engineering, Science Advances, and also has been honored with numerous awards, including the PMSE Future Faculty Award by the American Chemical Societies and the National Multiple Sclerosis Society Award. So without further delay, I'll just let Dr. Zhao to take the stage and share your research. So Dr. Zhao, please.
Thank you very much for the kind introduction. Let me share my screen. Is my screen sharing okay?
Yes, all good.
Cool. Thanks, Trey, for the introduction and also for inviting me to this great symposium. So yeah, I'm Shijie, and I'm currently just started my lab at the University of Washington. But today I'm going to share some of the work that have done during my postdoc, which is about some project strategies to deliver microbial metabolites for immune modulation. So I like to start with talking about these non-communicable chronic diseases, which we saw and are still on the rise, especially in the developed countries, and we also see such rise in countries such like China. Many of these diseases share some common properties that are related to our immune system. And also, this trend on the rise cannot be just simply explained by the genetic factors. Many of these diseases we saw that have been linked to many of the modern lifestyle factors. For example, the use of the antibiotics, the transition from the rural to the urban area, and also less exposure to the pathogens. And also people are now used to some fast food with the high fat and the low fiber diet. And but they actually really was some of the behind reason was the people found to be the altered commensal microbial biodiversity. So there's some more and more research are now going on to really investigate or look into how our commensal bacteria affect our immune system and then alter the really attributed to this disease progression. And one fact that we may not be aware of is that we actually have more bacteria reside in our gut than our total human cells, which also shows how important they might be or have a great impact on our human body. So what can this bacteria actually do? So it is actually a very complicated way that how those microbiome affect our human body. And here this simplified diagram showed one possible way that they modulate our system, immune system through the metabolites, which is produced by this microbiome, and how this microbiome modulate different organs and different systems in our body to affect how we respond, especially regarding to our immune system. So I'm not going to this in the details on how the microbial metabolites modulate the immune system, but what we were trying to do instead of delivery these microbial bacteria, which is more complicated, we want to ask the question that can we directly deliver these microbial metabolites as a therapeutic to modulate the immunity. So in our research, we are particularly interested in one small molecule called butyrate, which is a short-chain fatty acid that produced by the gut microbiome from the fermentation of the dietary fiber and the resistant starch. So it has multiple immune modulatory effect especially on the immune cells in the gut. So for example, it produces or expand the regulatory T cells, which is the cells that kind of induce the immune homeostasis in the body. And also it acts on the dendritic cells and the macrophages, which induce the tolerogenic effect in the gut tissue. And in addition, it also acts on the innate lymphoid cells that produce a cytokine called IL-22, which promote the gut barrier integrity to maintain the gut homeostasis. So because of these effects that people have found, it's really emerging to have explored the therapeutic potential of these small molecules, and people have really tried that in many different types of diseases. So one of the advantages of using these small molecules as a potential therapeutics is that they are naturally occurring in the body, which means that they are also generally regarded as safe. And it has already gone through some clinical studies which shows that there's no adverse effect and well tolerated in the body. However, it has not been successfully applied clinically due to some big challenges. For example, it has very bad taste and smell from these small molecules, and also it can be quickly metabolized in the GI tract, which also requires super high dose to achieve efficacy. So I'm going to talk about two strategies that we have developed here in the lab. So we're trying to what we're trying to do is to using these different drug delivery tools, both applied orally as the oral administration, to deliver butyrate through different ways that we can enhance the transferability into the therapeutic applications. So the first one is a polymeric mast cells that we are used to deliver the butyrate to the distal gut. And the second one is a prodrug with the amino acid called serine, which we conjugated to the butyrate to enhance its systemic biodistribution. So I will talk about these two strategies one by one. So the first one is a polymeric mast cells that we designed to specifically deliver this small molecule to the distal gut. So the idea is that we conjugate butyrate to the side chain of the polymer, and then this through the ester bond. And this polymer, which has which is a copolymer that have the hydrophobic chain with the butyrate conjugated, and also has a hydrophilic copolymer. So which allows this polymer to self-assemble into the mast cells. So so in that case, we were able to further protect the butyrate inside the core that can only release the butyrate once it reached to the lower GI tract. So we designed two different butyrate mast cells, which they share the very similar structure, but they also have the differences on their surface charge. So because of the different composition of the hydrophilic chain, we were able to produce one butyrate mast cell with the neutral surface charge, and the other one with negatively charged surface. So you can also tell from this characterization table that these two mast cells all have the diameter around 40 nanometers, but they have a different zeta potential showing the different surface charges. So the idea was to deliver the butyrate to the distal gut, where the most bacteria reside in the gut, such as the lower small intestine called ileum, or the large intestine colon. So in these studies, we first looking at where these mast cells release the butyrate in the mouse GI tract. So we took so we give the mice with these mast cells and took different compartments of the gut to look at the butyrate concentrations there. So we showed that actually, surprisingly, that the neutral charge particles release most of the butyrate in the lower small intestine, which is the ileum, while the negatively charged particles release most of the butyrate in the cecum of the mice. So they actually have a slight different release kinetics when we orally gavage them to the mice. So next we because the butyrate was known for its protective effect on the intestinal barrier permeability. So we want to see whether or not we can deliver the butyrate to the distal gut and also maintain its protective effect. So in this model, we given the mice with a chemical called dextran sodium sulfide, which can disrupt the intestinal barrier. And during that time, we also gave the mice daily gavage with either the control drug or our butyrate mast cells. So we've shown here using a fluorescent dextran, we are given orally gavage, and we measure the signals in the blood showing the leakage from the gut to the blood. And we've shown that the mice treated with our positive control drug or the butyrate mast cell drug has prevented this leakage and showing the protective effect on this intestinal barrier permeability. So with that, also some other immune characterizations, which I'm didn't go into details for that. We want to see whether or not we can use this to apply to treat some diseases, and in our case, food allergies, because it's highly relevant to the gut barrier functions as well. So in this case, we use a mouse model of peanut allergies in that we sensitize the mice first with a peanut extract and a adjuvant called chlorotoxin for 4 weeks. And the mice was also treated with the vancomycin to deplete the butyrate producing bacteria that we want to eliminate that inherent butyrate in the mouse body. And once the mouse gets sensitized or developed the peanut allergy, we treat the mice for 2 weeks with the tristatic gavage of either PBS or butyrate mice cells. And then we challenge the mice with the peanut to see the effect of our drug. So in this model or in any mouse model of food allergies, people use this gold standard temperature measurement to see the anaphylactic reactions to the food challenge. And in our case, when the mice given the peanut by IP injections. So when the mice experience the anaphylactic shock, they will experience the a drop in their body temperature. So in this case, we're showing that the mice treated with butyrate mice cells had barely no temperature drop compared to the mice treated with PBS, which experienced severe anaphylactic reactions upon the peanut challenge. And we also measured some serological readout, including the MMCPT1, which is a marker for the mast cell granulation, the histamine, which we all know is the chemical released from the allergic reactions. And this readout, we all see that the mice treated with butyrate mice cells had reduced responses there. And we also see that the peanut-specific IgE, which is the antibody that really drives the allergic reactions, got reduced from the butyrate mice cell treatment. And to have a better control, we also added the free drug called the sodium butyrate, given the same time, same dose compared to the butyrate mice cells. And in that case, we didn't see any therapeutic effect from this free drug, showing how important it is to deliver the butyrate to the distal gut to have this therapeutic effect. And we also look at the some immune cells in the gut draining lymph node. Here we look at the ileum lymph node and the colon lymph node to see how this treatment affect the immune cell some cell markers there. And here we are particularly looking at the CD86, which is the co-stimulatory marker showing the activation of these myeloid cells, or the MHC2, which is antigen-presenting molecules on these cells. So we have what we show here is that the mice from the mice treated with butyrate mice cells, we saw the significant reduction on these molecules, including the CD86 and the MHC2 in these on both dendritic cells and macrophages. So, so with that immune cell characterization, we also were curious whether or not with this treatment, by delivering these microbial metabolites, can also alter the microbiome composition in these mice. So in the same study, we collected the fecal samples before and after the treatment, and looking at the microbiome composition using a technology called 16S rRNA sequencing to look at how this microbiome changed from the treatment. So comparing the mice treated with either PBS or the butyrate mice cells, we saw these 5 bugs that got significantly changed. And we wanted to highlight one of the bug called Clostridium 14A, which is a very well-known bug that producing the butyrate in the healthy individuals. And we saw this bug was significantly increased by the treatment of butyrate mice cells. And this was encouraging because it means that these therapeutics could potentially also promote the increase of these bacteria that provided maybe we don't need to continue this treatment, and for super long term that this bug can be recovered and produce the butyrate by themselves. So we hypothesize there might be a positive feedback loop that when we deliver the butyrate, which is also known to induce the hypoxia in the gut environment, that promote the growth of these anaerobic bacteria. So just in a short summary for this project, that we deliver these, developed these mice cells that was able to deliver butyrate to the distal gut, and we show the improved integrity of the gut barrier, and it rescued dysbiosis, and also was shown highly effective in treating food allergies and the colitis in mice. Yeah, I didn't show the much details in the colitis model, but you can also find in our recent publications there. So that was the first story that we use these mice cells to deliver butyrate to the distal gut. So in the second part of my talk, I'm going to show these different strategies that we trying to apply in the delivery of the butyrate. And it is a very simple molecule that we develop using this amino acid called serine to enhance its bioavailability. So the idea here is to utilize the amino acid transporters in the gut epithelium and also in the blood-brain barrier, which our body uses to get nutrients in the tissues and cells. And we found that so we try to kind of hijack these transporters to deliver the butyrate to go through these to cross these biological barriers that bypass the quick metabolism in the gut. So we utilize this amino acid called serine, which showed was able to was present. The transporters for serine was present in many of these biological barriers. So by utilizing this amino acid conjugation to the butyrate, we measured the biodistribution of the butyrate after oral gavage, and measured the butyrate concentration in many different organs, including and also the plasma. And we showed that through this conjugation, the serine butyrate has dramatically increased the concentration of the butyrate in many of these major organs, and including the brain and spinal cord, which is normally thought to be very hard to reach from the free drug. So we directly just jump into some animal disease models, particularly because we show previously that butyrate can modulate the T cells, induce more regulatory T cells, and also modulate the myeloid cells, which are all play very key important in this autoimmune disease called rheumatoid arthritis. So this disease is triggered by the immune cells that attack the collagens or the some bone matrix there to induce the cartilage damage, and especially kind of mediated by the T cells and the macrophages in the joint. So we use a mouse model of the rheumatoid arthritis here. Basically, in this model, we inject the mice with these atherogenic antibodies that induce the arthritis in the mice. And we treated the mice with daily gavage of either PBS or serine butyrate to compare the therapeutic effect. And in this model, we here using some criteria to measure the clinical score by looking at the inflammation or swelling of their feet, and we show that the serum butyrate really are very effective in protect this the progression of the disease. And looking at some cellular readout from the draining lymph node, we show that the serum butyrate was able to increase the regulatory T cells, while it also decreased the pathogenic T cells. These are Rho gamma T positive CD4 T cells, or called Th17 cells, which drives the inflammation for these autoimmune conditions. And when we look at some other cell types, we also see these M2 macrophages, which is known for its anti-inflammatory effect, got increased by the treatment of serum butyrate. And we also see an increase of the IL-10 positive B cells. The IL-10 is also a non-cytokine to promote anti-inflammation in the tissue. So with the success for these preliminary studies on the rheumatoid arthritis model, we further look at into another more harsh autoimmune disease model called multiple sclerosis. So this is more like a neuroinflammation that the immune cells attack the myelin that's surrounding the neural tissues there. So in this model, we use a experimental autoimmune and encephalomyelitis called EAE to test our drug, the effect of our drug in these disease settings. So we give the mice with similar dose, but twice daily of either PBS or serum butyrate. And we also see that the serum butyrate was very effective in preventing the progress of the disease. And from these images showing the immune cell infiltration, we also see that the serum butyrate was protective to inhibit the immune cell infiltration into the spinal cord. And for several for the other immune cell characterizations, again we see some MMC2 decreased expression on the macrophage and the dendritic cells in the spinal cord, and also we see the reduced Th17 and Th1 responses in the spinal cord. Last but not least, we have consistently seen that the serum butyrate has increased regulatory T cells in the spleen and in the lymph node, showing that it's and in all these relevant lymph nodes, which is consistent with what we saw from the other disease model, that with the systemic delivery, these butyrate are really effective in inducing these regulatory T cells. So with all that, I'd like to just summarize these two projects that we either using these polymeric mice cells to deliver the butyrate to the distal gut, and we applied them in treating the food allergies and the colitis. And in the other story, we use these simple amino acid conjugation strategies to deliver butyrate systemically, and we applied them in the application of rheumatoid arthritis and multiple sclerosis. So with all that, I'd like to thank my postdoc advisors, Jeff Hubble and Kathy Nagler, and also these amazing graduate and undergraduate students who worked very closely with me during my postdoc for this project. And also, we just started our lab last month, and we are also continuing to investigate in the area of immunoengineering and also microbiome pharmaceutics. And if anyone is interested in our lab, also feel free to reach out to me. You can also send an email to me. And also, yeah, thanks for your attention. And also, I would like to thank again for this great opportunity.
Thank you so much, Shijie, for the wonderful talk. Beautiful works by using different delivery tools to shuttle butyrate to different place, and well demonstrating the delivery, the importance of delivery in treating different various diseases. So our second speaker is Dr. Feng from Indiana University at Bloomington. So he will talk about the automated tumor chips for cancer immunotherapy. So brief introduction about Dr. Guo. So he's currently an associate professor in Intelligent System Engineering Department at Indiana University, Bloomington. He received his PhD in engineering science and mechanics at Penn State and completed his postdoc training at Stanford University. He has published over 17 papers, including some high-impact papers in Nature Medicine, Nature BME, Nature Communication, PNAS, and so on. He's the recipient of various awards, including the prestigious NIH Director New Innovator Award, BP2, and the Outstanding Junior Faculty Award at IUB. So without further ado, I'll let Dr. Guo take the stage and share his research. So Dr. Guo, please.
Yeah, thank you. Okay, is that right?
Yes.
Okay, yeah, very nice. Yeah. Thank you so much for joining. It's a nice introduction. So today I would like to introduce our work on automated tumor chips for cancer immunotherapy. Yeah, maybe the topic is a little bit different from drug delivery, but we are developing devices for helping the cancer immunotherapy. So basically, we know the 2018 Nobel Prize has been awarded for the cancer immunotherapy. Yeah, we all know the immunocheckpoint inhibitors like PD-1 or CTLA-4 can significantly improve the T-cell toxicity, especially for treating the blood cancers, right? Yeah, this is something very famous now. And also, if you look into the cancer immunotherapy market, yeah, this is a super huge, right? The global market was about 85 billion, yeah, in 2021. Then it will reach maybe like 277 billion by 2030, yeah, with an annual increase about maybe 12.6. Then there are several outstanding challenges in the field. For example, the personalized cancer therapy, and also how to treat the solid tumor. Yeah, those are the problems. Yeah, we are engineers. We are thinking, yeah, how to address those problems. Then like for example, we want to look into this personalized therapy. If we look into the details for the companion diagnosis, yeah, there are significant heterogeneous response to the adjuvant therapy after the tumor surgical removal. Yeah, actually, it is challenging to choose the right adjuvant therapy for an individual after the tumor surgery. Yeah, this is a tremendous challenge for all the physicians. Then, if you really look into the current model, yeah, pretty much you can see patient-derived action graph (PDX). Yeah, and also the patient-derived organoid models. They are commonly used for the cancer companion diagnostics. Yeah, basically to predict which could be the best treatment for the patient, right? However, there are several challenges. For example, yeah, to have those models, normally we need a very long time. Yeah, for example, yeah, for the PDX, it normally will need 1 month, 2 months, something like that. If you want to do the patient-derived organoids, you may also need at least 2 weeks. During this lengthy model formation time, yeah, you will lose most of the primary cell types. For example, the myeloid-derived suppressor cells or MDSCs. Yeah, this is a challenge. So as a engineer, specific like a device engineer. So kind of what we think would be how can leverage our expertise to address this problem. So that is why kind of we are thinking using our strategy like acousophotic. Because this is my training background. Maybe I can introduce a little bit about this. During my PhD training, so we developed this acousophotic technology by integrating surface acoustic waves with microfluidics. So then we can have this kind of acousophotic device. Pretty much like this. It really have many kind of advantages. For example, it can be simply fabricated through the MEMS fabrication technology, and also it is a very simple, inexpensive setup, and also it can really handle cells and liquids with very high precision, even kind of down to single cell level, and also it is very versatile. It can be simply kind of adapted for many different translational applications. Then let's see how it works. So this is a typical kind of acoustic tweezer device. We have 4 sides of a electrode deposit on a piezoelectric substrate. Then we call those kind of deposited kind of electrodes called IDT, interdigital transducers. Then if we put on the radio frequency signal, something like this, then let's see what will happen. You see kind of the polysaccharide beads with a kind of a 10 micron diameter. It can be trapped into those kind of dottery patterns, right? So at the same time, it also can be lifted a little bit. You can notice from the defocusing to be well focused. From this video. Then how about if we put a phase angle between one pair of IDT? Pretty much you can see, kind of the trapped kind of polysaccharide beads array can be transported around X or Y direction. Then how about if we turn off the acoustic field, all the thing can drop into the substrate with well-maintained this kind of structure, right? So pretty much we demonstrate the 3D manipulation of acoustic tweezer device. Then the principle could be very simple. Basically we form this kind of 3D trapping nodes by kind of using this interaction of acoustic streaming field and also acoustic field. Then by precisely manipulate these trapping nodes, by tuning the frequency, power, everything, then you can precisely kind of manipulate particles or cells. And also we demonstrate we can do the single cell printing. For example, we can pick one cell, then kind of transport the cell to the desired location, then put it onto the substrate. By doing this, we can kind of print something like a 3D AT, which stands for the 3-dimensional acoustic tweezer. Then the advantage would be we can achieve a contactless, label-free, and highly biocompatible manipulation. It really could be used for many different applications. Then this is really my PhD work. I feel very elegant. Then when I really have my own group at IU Bloomington, kind of I was talking with my students. You see, can we really use these surface acoustic wave devices to address a problem? Then I actually my postdoc Zhao kind of he has a training on cancer biology, and also my students Zhao kind of he's trained on kind of a device side. Actually, kind of when we were talking, then both of them they thought, your device looks good, but maybe it's only good at handling kind of a single cells. Once we really go to the clinical sample, we may handle millions of cells. And also, we don't really prefer the closed chamber. Like you need to really have a microfluidic chamber. That is not a very convenient. Then they want to just make something very simple. Then they come out a very simple idea, just put a 4 side of PCT transducer, then also put on this petri dish. Then they can make a very simple device. Then they develop a new kind of acoustic cell assembly technique to address those challenges. Then we also demonstrate we can establish this kind of acoustical patient-derived cell cluster, APCC, within 2 minutes. Yeah, address a problem for the lung-seq culture, yeah, for the PDX and the patient-derived organoids, right? And also, we can study the tumor, T-cell, MDSC crosstalk. Yeah, since kind of we can rapidly form those cell clusters so that we can study everything kind of within 1 day. And also, luckily, the MDSC is a kind of lifespan in vivo is also something around 1 day. Then also we can test their response to different therapy within 1 day. Yeah, kind of yeah. This is looks good since the turnaround time only within 1 day or 2 days, and also the setup is very simple. Then later, kind of we are collaborating with Dr. Liang Chen from Brown University. Then we can collect some patient kidney tumor sample to test our device. Then we compare our 3D model acoustic cell cluster with 2D cultures. Yeah, pretty much we can see, yeah, we can preserve all the cell composition. Yeah, pretty much most of the kind of primary tumor cell types, like, yeah, since we have great interest for this MDSC, and also we can preserve very good cell viability. Then also, although we don't really have the kind of a tumor structure, but we can preserve the tumor microenvironment, like a cytokine and gene expression, everything, because we have very short turnaround time, only within 1 day, right? Yeah. Then after that, we also kind of try to manipulate the cell composition within this cell cluster. Yeah, to demonstrate the kind of flexibility of our model. That if you kind of depleting MDSC, you can see the tumor death increased, and also T cell cytokine secretion increased. If you deplete this CD4, CD8 T cells, you can see the kind of tumor dies kind of decreased, and also T cell cytokine secretion also decreased. Then we demonstrate this kind of MDSC can preserve their kind of original phenotype, like they can really suppress the T cell cytotoxicity, right? Then later we also try to test kind of target therapy, and also combine with PD-1 immunotherapy. Yeah, pretty much you can see through the combination, like couple this target therapy for MDSC and also PD-1 therapy. The combination therapy can really provide a better efficacy. Then we also demonstrate, yeah, like by kind of treating those cell cluster with different therapies, we also can kind of model or predict the therapy response. Yeah, we do hope this kind of a technique may kind of provide a potential kind of application for the clinical combination diagnosis. Yeah, then later we also develop other different types of devices for testing different cancer therapy. Yeah, then actually our final goal would be to develop a very simple and user-friendly system for cancer detection and treatment kind of prediction. It's pretty much like a coffee machine. Yeah, this is something ongoing. Yeah, then the other thing we are thinking would be like, yeah, current cancer immunotherapy are very effective for the blood tumors, but not kind of so great for the solid tumor yet. For the solid tumor kind of immunotherapy, yeah, kind of we are thinking about how to promote the T cell fighting with cancer cells in such a very complicated tumor microenvironment. So there are kind of two basic questions, like how could a T cell detect and kill the tumor cells? Yeah, We all know that CAR T, everything, right? So this is a main focus for the current field. Then the other thing would be how could T cell infiltrate into the solid tumor? This is largely understudied. Then my postdoc Zhao thought, yeah, this could be a good opportunity for us. Then we propose to screen immunocheckpoint inhibitor. Yeah, those are drugs for promoting the T cell infiltration. Then we all agree on that. Yeah, we know the kind of a T cell tumor infiltration is a very interesting and also very complex process. In the solid tumor, yeah, you see, we have this kind of CAF, we have this kind of cancer-associated fibroblast. Yeah, those cells will secrete kind of extracellular matrix, secrete the ECM, is like build a barrier to against the T cell infiltration, right? So this is a problem, and also, yeah, it also get involved with many different immunocells within the tumor environment. Yeah, the situation is very complicated. I was thinking, oh, yeah, we can make an in vitro model to study the dynamic interaction of T cell and also those tumor fluid. Yeah, I was also thinking, yeah, maybe we should really track the trajectory of the T cells. Maybe also at the same time we can really identify the T cell killing events. Yeah. So the idea is good, but John thought, oh, yeah, so maybe that is good, but he more want to focus on the biological function of the T cells. Yeah, actually, myself is more from engineering side. John is more from the biology side. We couldn't really convince each other. Yeah, because it is really hard to evaluate those cancer immunocheckpoints based on kind of one or two parameters. Yeah, we really have kind of a long discussion. Yeah, we somehow stuck there. Then actually my students come. Yeah, like a Hongwei. Hongwei thought, oh, yeah, it's good, but it's still hard to really understand this process. Can we really find something very simple? Then Hongwei thought, so we really use a kind of a TIL map. Yeah, TIL refer to the kind of a cancer infiltration lymphocyte. Yeah, within a kind of a clinical tumor, you will have all the kind of T cells distributed. Yeah, into a slice. It's something like a map, right? So can we really use the TIL map? Yeah, since within the TIL map, we have the infiltration depth, we have the cell number, we also have the cell spatial distribution, all the information. Yeah, I agree. This is something similar to my idea. I think, yeah, I thought this is good. The whole way also come after the other thing. Yeah, it's also hard to evaluate. Yeah, for example, even the TIL map, how can you tell this is a good kind of drug or not? Yeah, then kind of he was thinking, should we really use the patient survival time as an outcome? So we just consider this kind of TIL map kind of correlated to the patient survival time. This could be a really good strategy for the drug screening, right? Yeah, I thought this is good. Finally, Hongwei convinced me and also convinced Zhang. Then we are also looking into the literature. Actually, the clinical data indicates the longer survival time is always associated with a higher kind of TIL infiltration and also with a certain pattern of the TIL maps. Yeah. Then my student Hongwei trained a deep learning algorithm. Yeah, we call it a TIL score analysis. Yeah, to project the TIL map with the survival time using those clinical data. Then basically we can using this kind of trained deep learning algorithm to generate a TIL score. Yeah, basically the TIL score is more like a pseudo survival time, right? But we can use this TIL score to evaluate each kind of compound. Yeah, once we get the each heal map or tumor image from the patient, yeah, we thought this could be something very exciting. Then we can address a problem. Then, if you really think about the drug screening part, actually, current tumor organoid are still suffering from the problem. For example, if we do the kind of a tumor organoid culture within the petri dish, yeah, actually, kind of yeah, they are rotating. You couldn't really get a very good T cell and tumor spheroid interaction imaging from there. Of course, yeah, we are device guy, we are microfluidic guy. We are thinking, oh, can we really make a high throughput microfluidic platform to trap those kind of tumor spheroid? Then later we can put in the T cells. Then we can just do the time lapse imaging for all the T cell tumor spheroid interaction dynamics. Yeah, I thought this is good. And also, we also want to combine with this kind of a drug score system. Then we just try to combine two things together to make an automated microfluidic screening platform. Then we aim to discover some new therapy that can enhance T cell infiltration. Then, yeah, we just showcase, yeah, we really can generate those kind of a tumor kind of a surface. Yeah, first we see this cancer cell onto this kind of a pillar lattice design. Then later sit on the calf cancer-associated fibroblast. Yeah, then they can form this kind of a heterotopic kind of cell surface. Yeah, then the advantage for this design would be we can immobilize those tumor spheroid that later flow in the T cells and do the imaging, right? Yeah, basically we also can form a very uniform tumor spheroids with those kind of a core shell structure. This is also something really good for the drug screening. Yeah, since we always want to generate a very uniform tumor spheroid, right? Yeah. This will bring us advantage to the current kind of well-played based tumor organoid or tumor spheroid culture. Then we also demonstrate we can track the T cell infiltration dynamics. Yeah, you see from here. Sorry. Yeah, this is the T cells. Yeah, it can infiltrate into the tumor. It also can show the kind of killing event in a dynamic manner. So let's say again. Yeah, enter into the tumor spheroid. Yes, the first killing. Yeah, then the second killing. So yeah, basically we can track the dynamic T cell tumor infiltration and kind of cytotoxicity together, right? Based on this kind of yeah, we're thinking, can we really do the drug screening? We just get a very kind of a small drug library, has a 141 compounds. For example, if we pick up one compound, we put it into our system. Yeah, we have thousands of tumor spheroid there. Then we can get the kind of image from there. Then we can extract the kind of TO maps. Then go through our deep learning algorithm. Then we can get a Q score for kind of each image. Then if we average this score, we can get the drug score. Yeah. Then by doing this, we can generate all the drug scores for the whole library. Yeah, luckily, we got a compound, GSK, LSD1, that can significantly enhance the T cell infiltration. Then later, we want to validate this compound using animal. Then kind of after kind of a tumor implantation, then we test the animal with kind of anti-PD-1, JSK, and also their combination, and also the PBS vehicle. Then we can see the TO number and the infiltration depth results pretty much like this. Pretty much we can see the GSK is better than the PD-1 in promoting the kind of TO infiltration. And also you see the tumor results and also the TO infiltration results also showed the combination, the GSK and the PD-1 can provide a better efficacy than only PD-1 or only GSK. So with entry, maybe the combination therapy is better than GSK or PD-1 only. Yeah, it may be a good kind of a candidate for clinical study after the toxicity testing. Yeah, the summary. Yeah, basically we demonstrate some tumor tips for testing and predicting cancer immunotherapy within 1 day, and also we develop an automated platform for screening kind of immunotherapy against the solid tumor, and also we found the combination like GSK and PD-1 may be good for improving the current solid tumor therapy. Then I want to acknowledge my group member and the collaborators. Yeah, actually my student and postdoc, they did all the work. Yeah, I also want to give a special kind of thanks to my postdoc Zhang and also my students Hongwei and Zhao. Yeah, they lead those projects I present today. I also want to thank my collaborators. Yeah, they significantly promote our research. I also want to give special thanks for my collaborator Liang Chen, yeah, Zongmin, and also Xin Lu. Then other than that, I also want to acknowledge my research funding. Then thank you.
Thank you so much, Feng, for giving a fantastic talk. So by presenting an elegant model to study the immune and the tumor crosstalk. All right, so our next speaker is Dr. Zongmin Zhao from University of Illinois Chicago. So he will talk about cell-inspired strategies for precision tissue targeting and immunodelivery. So a brief introduction about Dr. Zhao. So he received his PhD degree in biomedical engineering from the Virginia Tech in 2017. And later on, he did his postdoc training at Harvard University with Professor Metabolites. And to date, his research effort has led to over 50 published articles, such as in Cell, Nature Biomedical Engineering, PNAS, Science Advance. And his work was also recognized by numerous awards, including the R35, NIH Maximizing Investigator Research Award, MIRA Award, and he is currently an associate editor of the AT Journal Bioengineering and Translational Medicine. So without further ado, so I'll let the Zongmin take the stage. So Dr. Zhao, please. And then you unmute.
Okay.
Yeah.
Can you hear me now?
Yes.
Okay, cool. All right. Thanks, Shijie, for the introduction, and really appreciate the opportunity to actually share some of the work that I did over the years. And the topic is basically cell-inspired strategies for precision tissue targeting, and also some immuno-delivery work. And as the engineer or as the drug delivery people in the field, actually the long-standing challenge for the drug delivery field is the biological barriers, and these biological barriers has been there, because the body is very smart, and if we dose a drug to the body, we have to overcome these several biological barriers. Especially for the immunodelivery, we also have additional layers of biological barriers. And one of the most distinguished biological barrier is basically this intravascular barrier. You have a lot of innate immune cells in the bloodstream or even lining on the organs, for example, macrophages, and they take up these drugs or even drug carriers sequestered there, so they cannot go to the drug target. So basically, over the decades, there are many efforts have been done in the field developing different drug carriers to overcome these barriers, and that including nanoparticles And the one actually very exciting field or actually emerging field is to actually basically taking inspiration from the biology to overcome these biological barriers. And one such example is from the body own cells. The reason why we think that the cells can be a good drug to carry is that as compared to synthetic materials, our body's own cells, they are natural in self, so they cannot be easily clear out from the body quickly. And apart from that, another distinguishing feature of cells is that some cell types actually have the active transport properties. For example, macrophages, neutrophils, or some other white cells, they can basically easily transport to tissues that undergo the infection, inflammation, even cancer. So this really inspired the field actually, how to leverage these cells as a living drug carrier. As you can see, over the past 10 years, the field actually has evolved a lot. There are many breakthrough technology out there, including, let's see, using red blood cells, monocytes, and pioneered by Samir Mitragauchi. I did my postdoc with him. I will introduce some work that I did with him, and also, let's see, platelets. Chen and Dr. Gu pioneered this responsive platelets, and most recently, also programmable bacteria as a platform for target delivery to tumors or some other immune pathologies. And some of the work that I did over the years is basically to take the inspiration from the red blood cells, and we developed this red blood cell based technology to deliver drugs. Especially, we are interested in to deliver nanoparticles. So what's unique about red blood cells? So as we know, red blood cells, one significant feature is that they can circulate very long in the blood. So basically, they have a very long lifespan, 120 days for human red blood cells. And this is because red blood cell surface actually express some self receptors. Let's say they express CD47. So this will make the red blood cells invisible to the macrophages, and so they will not be phagocytosis, so will not be cleared out. Apart from that, as we know, the major function of red blood cells is for oxygen transfer, and this is achieved by red blood cells going through different tissues of the body. So basically means red blood cells have access to most of the tissue of the body, so we can leverage red blood cells to achieve delivery to many tissues of the body. So I think as I mentioned, we are interested in delivering nanoparticles, but the first question we want to figure out is that how we actually can place particles on red blood cells. There are definitely many strategies you can do. Let's say if you can conjugate them using chemical conjugation, many strategies have been reported, or you can rely on the electrostatic interactions. However, we figure out that we can just simply mix nanoparticles with red blood cells, and this will achieve efficient binding of these particles to red blood cell surface. And the mechanism for this binding relies on the non-covalent interactions between the nanoparticle surface and the red blood cell surface, including the electrostatic interactions, hydrophobic interaction, and also hydrogen bonding. And back in about 10 years ago, Samir's group actually they first created the formulation, and they injected this formulation into mice. And they've actually found very interesting results. Let's see. The first very interesting result is that by putting this nanoparticle onto red blood cells, they can significantly improve or extend the circulation time of the nanoparticles. As you can see here, if you inject free particles, they got cleared very quickly. However, when you put this particle onto this red blood cell surface, you can significantly extend their circulating time in the blood. And what's even more interesting is that beyond changing the pharmacokinetics, you can also change the biodistribution of the nanoparticles. For example, as you can see here, when you inject free particles, basically majority of particles go to the liver and spleen because you have macrophages there. However, when you place this nanoparticle onto red blood cells, basically we can shift the delivery to the lungs. And in the meantime, the carrier aerosols that remain in the blood basically means these red blood cells really hand off the particles to the lungs, and then themselves remain circulation in the blood. So the mechanism for this red blood cell-based long-targeted nanoparticle delivery is really relies on the biophysics of red blood cells. So the major function of red blood cells for oxygen transfer, and this is achieved by red blood cells squeezed through the capillaries in the lungs. So we can imagine when you have nanoparticle on the cell surface, when they go through the capillaries, these particles really get mechanically sheared off and transported to the lung endothelium. This is the reason why we can deliver particles to the lungs using this technology. And by the way, this technology is called aerosol hitchhiking, or EH. Okay, now we know this technology can deliver particle deliver drug to the lungs, and obviously we'll have a lot of implications. And the first implication, which is very obvious, is that we can target some lung disease. And the first indication we were interested in was actually lung metastases. So what is lung metastasis? Lung metastasis is basically, the primary tumor metastasized to the secondary organ, and in this case, is the lung, and it's advanced cancer stage, and it's actually very prevalent. Over 30 to 55% of patients who have advanced cancer stage actually have lung metastases. So currently, there's no specific treatment for lung metastases, and it is treated the same way as the primary tumors. Chemotherapy is one of the options, but the efficacy is always limited because, again, we are facing a challenging delivery problem. So the accumulation of the chemo drugs in the lung metastases is really limited. So to solve that challenge, basically, we redesigned this aerosol hitchhiking, developed this aerosol library chemotherapy, which we call ELECT, for basically, trying to enable chemotherapy for treating metastases. And the idea is pretty simple. We use these red blood cell deliver particles to the lungs, targeting the metastasis sites, and the particles will release drugs. And in this case, actually, we use the immunogenic cell death inducer, doxorubicin, and these chemo drugs can actually take out the tumor. As you can see here, we basically did the first experiment. We put nanoparticles, we loaded the drugs, doxorubicin, into nanoparticles, load them onto red blood cells. As you can see here, they actually bind to red blood cells very well. And then we tested if we can actually deliver these particles, deliver the drugs to the lungs, and we basically did a biodistribution study in the lungs, in mice that had lung metastases. As you can see here, by using this red blood cell hitchhiking approach, ELECT approach, we can basically achieve more than 15-fold enhancement regarding the drug accumulation in the metastases. And although not all the drug particles were in the metastatic nodules, but actually a substantial portion of the particles were colocalized with the lung metastatic nodules. And then we did an efficacy study, and it's actually pretty exciting. A lung metastasis model, and this is a very challenging B16 model, and we can actually extend the survival time by almost a month. And so as I mentioned, chemotherapy is good because it can actually delay the growth of tumors, but in most cases, it cannot completely cure tumor. And over the years, or at least over the past 10 years, immunotherapy as a field has, bring about a lot of exciting results. And one exciting thing about immunotherapy is that you can achieve tumor-free survival. And there are many different, actually at least 5 different classes of immunotherapy that have been approved by FDA, including the 3 ones that are listed here: immune checkpoint inhibitors, CAR T therapy, and cancer vaccines. And although we have achieved some exciting results in the clinic, but the current challenge for the cancer immunotherapy is that there is only limited success in solid tumors. For example, CAR T therapy is effective in liquid tumors, but they are not quite effective in solid tumors. Another challenge is that in advanced disease or poorly immunogenic tumors, like in metastatic tumors, the efficacy of this cancer immunotherapy is not outstanding. To solve these challenges, we basically actually going back and we rethink lung metastasis, releasing rethink the metastasis. So at the first glance, metastasis is actually a bad news, right? Because it's really hard to treat. However, when you rethink about metastasis, we think it actually represents opportunity for cancer immunization. So what I mean by that is you can imagine when you have tumor cells metastasize from the primary tumors to the second organ, actually these tumor cells got exposed, and you will have the opportunity to leverage the immune cells to attack these metastasized tumor cells, induce immune response. And however, there are quite a few challenges to leverage these metastases for effective cancer immunization. The first challenge is how to actually catch this early metastasis stage is very challenging. Second, how to actually deliver immunotherapies to be co-localized to these hard-to-reach metastasis sites is also challenging. To solve these challenges, we found that iris hitchhiking actually have opportunities there. And basically, as you can see here, in some of the work that we did before, we developed the iris suicide anchored systemic immunotherapy, easy for short, to leverage these metastases for effective cancer therapy. And the idea here is pretty straightforward. So basically, we load immunotherapy into the red blood cell hitchhiking or iris hitchhiking platform, and in this case, we use a chemokine, which is a compound that can actually attract immune cells. As you can see here, the idea is to put these particles onto red blood cells. We call these particles immunobate, and we can deliver these immunobate particles to be co-localized with the metastasized tumor cells in the lung endothelium. And the release of these chemokines can actually trigger a immunocascade and enhance the immune cell infiltration to the metastatic tissues and also generate the immune response. As you can see here, as a proof of concept, we generated these immunobate particles, place them on red blood cells, and they can bind to red blood cells very, very well. And we did some biocompatibility study and found that actually at a certain ratio, let's say below 1,000:1 ratio, 1,000:1 nanoparticle to red blood cell ratio, we will not induce significant damage to red blood cells. And this is quite important because subtle damage to red blood cells will actually direct delivery to other organs, which I will introduce later. And using this easy approach, we can deliver these immunobate particles to the metastases at a very high efficiency. We can achieve over 25-fold enhancement. And these immunobate particles are quite nicely co-localized with the metastasis nodules, and either inside nodules or they are in close contact, close proximate with the metastasis nodules. And this accumulation of these immunobate particles actually bring about a few immunological consequences. The first immunological consequence is actually the restoration of this chemical gradient. So as you can see here, in one of the first in vivo studies we did were that we checked the chemokine gradient in the lung metastasis with development. As you can see here, with the development of the lung metastasis, this chemokine gradient, especially this CXCL10 chemokine gradient, actually gradually lost. However, when we dose this easy, as you can see here, we give by only giving a single dose of this easy, we can actually restore this chemical gradient and sustain this gradient for at least 48 hours. And this restoration of the chemical gradient can enhance the infiltration of several classes of immune cells, as shown here. These are not all the immune cells that we found dramatically changed, but some of the immune cells that have the most dramatic change. For example, following the treatment by this easy approach, we can have Th1 cells, CD8 cells, effector CD8 cells, and also NK cells more infiltrated to these metastatic lungs. And this immune cell infiltration actually led to some therapeutic efficacy and in controlling the local tumors. As you can see here, basically following the treatment by this easy approach, we can significantly inhibit the progression of these lung metastases, and also we can achieve in some of the mice, over 25% mice, we achieved their survival. And beyond 40 days, this was quite challenging in this 41 model, which is a challenging breast cancer metastasis model. It was even more interesting that this EASE approach not only modulated the local environment, it also generated the systemic immune response. So we basically first checked the local DC activation in the metastases, because DC activation can actually bring antigen to the lymphocytes and generate immune response. As you can see here, following the treatment by EASE, basically we actually have more activated disease in the lungs. And then following treatment by EASE, we also checked the immune memory by checking the memory T cells. And we found that this EASE approach can also result in a higher number of memory T cells in the blood and also in the spleens. And then the following up rechallenge study also showed that, if you can see here, we basically rechallenged the mice with a second tumor, and following treatment by EASE, and we can actually suppress the growth of the secondary tumors. Basically means we generate a systemic response which can control the systemic tumors. As I mentioned before, the initial idea of using this aryl hitchhiking was to basically modulate the macrophages in the bloodstream, or modulate the innate immune cells in the bloodstream to actually bypass this immunorecognition of the nanoparticles. And this actually is very useful for modulating the immune response, or for improved delivery of very immunogenic therapies. And one such therapy is actually AAV-based therapy. Adeno-associated virus is a viral vector-based gene therapy, and this is a very promising viral vector-based therapy in the clinic. And currently, there are over 200 clinical trials, active clinical trials, for treating different diseases. But the challenge for AAV-based gene therapy is that it's very immunogenic. So basically means if you give the patient a single dose of AAV, they will develop a very strong immune response. And if you will not be able to give the second dose, because they will be neutralized by these neutralizing antibodies generated in the first dose. So to solve this challenge, we think actually this aryl hitchhiking actually have an opportunity there, because as we show in our previous results, we can actually bypass some of these innate immune responses. Another question, can we actually use this aryl hitchhiking to overcome this existing or reduce the primary anti-AAV immunity to enable this redosing? To answer that question, basically, we came up with an approach to figure out how we can place these AAVs on red blood cells. It's not simple, because we figured out that if you simply mix AAVs with red blood cells, they will not bind to red blood cells. And we came up with a chemical-based approach. It's a polyphenol-based approach that can bind these AAVs to the red blood cells very efficiently. As you can see here, these tiny particles are AAVs. They are sitting on the surface of these red blood cells. And then we designed a kind of simple experiment to figure out the redosing potential. As you can see here, basically we give 2 doses of AAV to the mice. In the first dose, we give them AAV vectors encoding GFP. And the second dose, we give them AAV vectors encoding luciferase. And the expression of this luciferase really is an indicator of the redosing potential. As you can see here, if we give 2 doses of free AAVs, basically the luciferase expression significantly drop. However, when we give the mice red blood cell formulations, we can restore some of the expression of the luciferase, although not to the full potential, but at least to some, as compared to free AAVs, we can enhance the luciferase expression in the lung tissue. So what about the mechanism? So initially we thought probably because we reduced the production of AAV antibodies. However, this was not the case, because as you can see here, actually in these different treatment groups, anti-AAV antibody titer is pretty similar. And then later we did a simulation study and basically simulated the binding kinetics between the AAV and the neutralizing antibodies, either in the free form or hitchhike to the red blood cells. And we found that by placing these AAVs onto red blood cells, we can basically delay the binding of these neutralizing antibodies with AAV vectors. Basically means we created a time window for these AAVs getting delivered to the target tissue. So as I mentioned before, the most distinguished property of red blood cell is is their lung circulation. However, in recent years, the field actually figured out that red blood cells actually have some innate immunological functions. This is probably surprising, but it's actually especially useful in red blood cell to clear blood pathogens. For example, if there's a pathogen invasion into bloodstream, red blood cells can actually kill some of the pathogens by oxygenation, and these dead pathogens can bind to red blood cell surface and got handed off to the immune cells in the spleen and liver and induce immune responses or got cleared from these immune organs. The mechanism for this process is not clear, but some preliminary data in the field showing that this is because of the transient damage of the red blood cell membranes. And the transient membrane damage will increase the crosstalk between these pathogens and the immune cells in the spleen. To mimic that process, we basically developed a second version of this aristotle hitchhiking. And in this version, basically we place some hard particles and we place more particles on the red blood cell surface. And by doing that, we basically achieve two purposes. First, we can make the red blood cells more rigid. And in this case, if they are more rigid and they are resistant to the lung shear stress in the lungs, so particles will not be easily shed off. So you can bypass this lung delivery. And the second consequence is that we can induce this transient membrane damage. And this will increase the crosstalk between these particles and the immune cells in the spleen. And as you can see here, in this second version of this aristotle hitchhiking, we can basically shift the delivery to the spleens and then bypass the lung delivery or reduce the lung delivery. And when we put the antigen on the nanoparticle surface, we can induce the immune response by targeting the immune cells in spleens. And in a lymphoma model which express ovalbumin, we can reduce or we can actually achieve similar efficacy as compared to some clinically relevant adjuvants, let's say CPG. So yeah, I mean, I have spent quite a few time to discuss this red blood cell hitchhiking. Actually, this aristotle hitchhiking can be applied to other cell types. And in some of the work in collaboration with Junlin group from Sichuan University, we developed this. We extended this concept to other cell types, basically using a polyphenol-based approach. And as you can see here, by using this approach, we can basically engineer different mammalian cells, including monocytes, macrophages, T cells, natural killer cells, dendritic cells. And one such example to showing this approach can actually achieve delivery efficiency, especially for targeting for immunodelivery, is that this approach can actually enhance the delivery of PD-L1 antibodies by hitchhiking or coating this PD-L1 antibody onto macrophage's surface. So macrophage has that intrinsic capability to traffic the tumor tissues, and this is because the expressed chemical receptors can sense the chemical signals and then traffic the tumors. And as you can see here, by using this polyphenol approach, we can efficiently coat this antibody on the macrophage surface. And in tumor-separated model in vitro, we see some very striking results. As you can see here, when you only treat the tumor spheroid with free PD-L1 at the center of the tumor spheroid, you can now see a lot of signal. However, when you place this antibody onto this macrophage surface, you can basically see a lot of signals in the center of the tumor spheroid, meaning you can improve the penetration of this antibody to the tumor spheroids. And in vivo, we also see enhancement of the PD-L1 efficacy by placing them on macrophages. And currently, there's some ongoing work in our lab that is currently basically trying to to modulate different immune environment or other immunodelivery in other diseases, for example, acute injuries, autoimmune disease, and metastases. So as I have discussed a lot, so as a researcher working in this cell-based delivery, a lot of people working in this area, including some panelists like Chen and myself working in this area. So basically, the question we are trying to answer is that how can we actually develop the next generation cell-based carriers, especially for immunodelivery? And the first generation basically will leverage the body's innate immune cells. However, the efficacy or their property is not ideal. And the second generation really involves a lot of efforts from different fields, especially from the cell biology and immunology, better understanding of these properties of cells. Also from the engineering field, we work in genetic engineering, epigenetic modulation, biomaterials. That's what we do for bioengineers, and ideal next generation cell-based carriers with a lot of properties. But basically, the overall goal is to increase the precision. Okay, to really find a place where to deliver, and the second, how to actually leverage the behavior of the cells to beyond delivery and achieve therapeutic efficacies. Okay, with that, I would like to just acknowledge, some, the support over the years, my current group members who work hard to trying to push some of this related technology to next stage, and also to my, postdoc advisor, Samir, who have been very supportive to some of the rebel cells that I introduced. Okay, with that, I'll happy to take any questions. Again, also, we are also recruiting postdocs. If you are interested, feel free to reach out by email, and we are always happy to discuss with you. Okay, cool.
Yeah, thank you so much, Zongmin, for the fantastic talk. Elegant work on showing how to leverage red blood cells for delivery immunotherapeutics. So I think now it's time to enter the next chapter of our webinar panel discussion. So first, we can go through the Q&A session. So maybe our two panelists, Sichen and Guojun, you can ask any question to our speakers. So I would like to start with our graduate student trainee, Sichen. Sichen, do you have any question for our three speakers?
Yes, hi, I'm Sichen from University of Wisconsin-Madison, and thank you for your excellent lectures from all three professors, and I learned a lot. Yes, and I would like to raise a question for Dr. Guo. He has mentioned the acoustal fluid discs for drug screening, especially for the immunotherapy. And I was wondering how well it's mimic the tumor and what's the limitation and advantages, especially when comparing to the traditional 3D organoid for the drug screening.
Yeah, thank you, Sichen. Yeah, very nice question. So yeah, I would see in the talk, actually, we may discuss the two different type of devices. The first would be the acoustic assembly cell cluster. So within this cluster, actually, the main advantage would be the turnaround time only within 1 day. So we can preserve most of the cell types from the original clinical tumor. Yeah, this is an advantage, but the problem would be we couldn't really preserve the original tissue structure. Yeah, this is the problem. Then for the next kind of type of device would be the pillary-based tumor spheroid here. Yeah, to really mimic the tumor kind of this kind of soma barrier. Yeah, we got this tumor core and also the kind of calf. cancer-associated fibroblast kind of shell. Yeah, we can form this kind of core shell structure so that we can mimic the kind of ECM barrier, so that we can test how the T cell penetrate. Yeah, here we have the physical barrier. This is good, but again, so we are missing kind of most of other immunocells. For example, here we only have within the simplified model, we only have kind of CD8 positive T cells, but we don't really have other kind of immunocells. Yeah. So eventually, the goal would be how can we make a reasonable, simple, and complex in vitro model for the high throughput drug screening. But again, we need to balance. Basically, we need to really think about what could be the best phenotype we are targeting on. Then we can make a decision to balance. Yeah, let's see. Thank you.
Thank you for your answer. And maybe I have a follow-up question. You have mentioned the CAF, cancer-associated fibroblast. And also, I noticed you mentioned the tumor microenvironment. And I was wondering how do you vision the application of your acoustic fluids especially in the tumor microenvironment related immunoengineering?
Yes, thank you. So yeah, certainly for the acoustic cell cluster approach, yeah, again, so main advantage would be this kind of a 3D cell sampling and also very short turnaround time. So yeah, actually, the main kind of focus or potential for our this kind of work would be for the kind of personalized medicine, like a personalized therapy. For example, yeah, we may have very huge patient-to-patient variation. Yeah, we may can get the clinical sample, clinical tumor, then try to assemble it with our acoustic device, then try to test the different immunotherapy. I think that would be a very good target for us at this moment. Then the other thing I'm thinking maybe also could benefit us to study, to understand the very complicated kind of cell crosstalk, especially among different kind of cell types like a cancer, CAF, and also different kind of immunocells. Yeah, that is something pretty much I'm thinking now.
Thank you.
Thank you.
Probably Guojun.
So, first of all, very glad to be here and to learn such fantastic new research area from those three very fantastic scholars. So, maybe I will take this opportunity to ask two questions for the other two speakers. So the first one is for Dr. Zhao. So I noticed that while I think it's a very simple platform, but it can achieve so many functions in terms of treating different diseases using this mice cells, right? So I noticed that when you introduce your work, you have a PBS as a control group, but do you actually compare to if you use the free drug like the butyrate, is that right? So as a control to what would be the main advantage of using of nano platform, or in terms of the oral delivery, would you consider the microparticles as an alternative too? Yeah, thanks.
Yeah, thank you, Guojun. Yeah, that is a very good question. Yeah, we do have a control for the sodium. So we use the salt butyrate called sodium butyrate as a control. So so we didn't see any therapeutic effect in our food allergy model there compared to the effect we saw from the butyrate mice cells. But the one big thing is that when we dose the mice with the free drug, it's mostly just consumed in the lower GI tract, or mostly even just in the stomach. And it's not gonna be, and it's got very quickly metabolized there. So not going to the systemic circulation and not going to affect the immune cells or the barrier functions when we just giving them the free drug. So maybe some people have really also done with just giving the mice with drinking water for really long time, which they see some kind of effect. But our strategy is to really efficiently deliver them to the lower GI tract so that they can really have those effects there with the controlled release in the lower GI tract. And also what we see is that when we deliver these butyrate in the lower GI tract, where most of the microbiome are reside there. So what we saw is that they can also actually modulate the microbiome compensations there by promoting more healthy microbiome, which are those butyrate producing bacteria in the lower GI tract.
Cool. So thank you very much for your additional information. Maybe another question for Dr. Zhao. So maybe it's the question is about a very versatile platform too, using this red blood cells to deliver the nanoparticles. So actually, two quick questions. So when you use this, how do you think that how uniform this platform can be when you prepare this drug, like attach or platform? And also another quick question is, so do you think this can be also so mostly I think in your platform is about nanoparticle space strategy, right? So do you think that this is an advantage, while there are advantages over the microparticle based platform?
Thanks for the awesome questions, Guojun. And to answer your first question, that's a very good point. So basically, for the first version of this technology, there's some heterogeneity there, because if you look at the SEM images, you will find that some cells actually have a lot of particles, some cells don't have a lot of particles. So basically, currently, actually, there's some ongoing work to develop microfluidic-based approaches. We're trying to automate the fabrication of these formulations, like using these microfluidics to actually reduce heterogeneity. And hopefully, the second version we actually can achieve like most of the red blood cells have like equally number, or at least close number of particles. So that's the answer to your first question. Second question, that's a very good point, because especially for the lung delivery, so as we know, some big particles may stuck in the lungs. And but to my perspective, I think the advantage or the beauty of this red blood cell hitchhiking is that really we are not limited to the size of the particles. Basically, you can put different size particles onto these red blood cells, or not even nanoparticles, or even let's say some naturally occurring with some particle shapes. Basically, can actually extend the versatility, the things you can deliver. And the second thing is that microparticles, they can stuck in the lungs, but actually they cannot efficiently cross the endothelial barriers. So by using this red blood cell approach, we can deliver nano-sized particles, and then by taking advantage of nano-size, they can actually penetrate depending on your indications. So these are two advantages I can see from the technology of the red blood cells compared to microparticle technologies.
Yeah, thanks.
Thank you. So I have one quick question for each of our speakers. The first, to our first speaker, Shijie. So how do you control the release of the butyrate in the distal gut?
So the strategy that we use is just a simple ester bond. So it's basically a conjugation that can only be cleaved by those enzymes and even bacteria that got enriched in the distal gut. So when the formulations that we also include is the micelles, which kind of encapsulate those conjugated butyrate in the micelle core. So we noticed that it's actually bypass the stomach, which is acid, could be also cleave those conjugation, but we actually protected the butyrate, those conjugation from the cleavage from the stomach. So it reached to the bypass the stomach, go to the the distal gut where there are more enzymes and more bacteria there, and the butyrate got more cleaved at that side.
Thank you. So my second question to Dr. Feng Guo. So you showed elegant work in building this acoustic microfluidic device to assemble different cells into mimicking the tumor microenvironment. So I'm just wondering, is there any limitation of your device to assemble how many cell types into this spheroid? Or is there any limitation to the size of the cells? For example, you assemble like tumor cells, CAF, and the immune cells into one spheroid. Have you thought about encapsulate or incorporate bacteria into this tumor microenvironment? Because bacteria is much smaller than the tumor cells and the other immune cells. Your device can still well enough to capture or to assemble this bacteria into your overall microenvironment?
Yeah, thank you, Chen. Short answer would be yes. And also, yeah, because based on the physics of acoustic, we will generate those kind of acoustic field and also acoustic streaming field, combine two field together. Yeah, then they will push everything together into a cell cluster. So yeah, it doesn't matter for what's a cell type. They will just push into a three-dimensional cell clusters. Yeah, actually for the bacteria cells, yeah, they are small. Yeah, but we also did something to cluster the bacteria cells together. Yeah, once you mixed with kind of big cells like tumor cells, yeah, kind of MDSCs, yeah, and also combined with kind of those bacteria cells, yeah, that would be very similar. You can push everything there. And also, other than this, would be think about kind of cell number per cluster. Yeah, by tuning the density, we also can achieve that. Then the other thing we are using would be we want to, since we only turn on the acoustic for less than 1 minute, then we really want the cells maintain in a 3D cluster. Then we also put in some kind of biomaterials or gels. Then those materials can be solidified during the acoustic treatment time. So after acoustic treatment, so everything can be assembled into the cell cluster. At the same time, you also some kind of immobilize everything there. Yeah, that would be really a good model for you to study the cell crosstalk. Then the limitation would be like I said, so it couldn't really fully capture the original tumor structure. That would be a problem. But again, so we do observe the T cell, they can freely travel within those cell cluster. That part is fun, but we won't really have the vessel structure or other well-defined CAFs created ECM barrier. We won't really have those things.
Thank you. So that's the question to Zongmin. I was very interested about your long-selective delivery by the red blood cells. I'm just wondering the mechanism for the drug release or the particle release. So when the red blood cells pass through the small capillary of the lung epithelial cells, so is the particle just get off the bus, get off the red blood cells and stuck into the epithelial cells, or is the drug squeezed out when they pass through these small capillary?
I see. That's a very good question, Shijie. We don't have direct evidence to. Okay, let me see. We did some body screen study. Basically, in this body screen study, we labeled the particles with dye, and then we found that we also tracked the red blood cells. Basically, particles get to the lungs, the red blood cell not. So basically means these really only the particles actually to the lung endothelium. But as you said, it's like leaking out of the drugs that we are not sure. But I believe probably not because of that, because some of the particles that we use, let's say PRD particles, they're pretty hard. So they are not soft enough to actually have the drug leak out from using that 6 Pascal shear stretch in the lungs. So most likely it's just particles, accumulated in the endothelium and then transport to across the endothelium and the drugs released from there.
Thank you. So thanks all for your outstanding talk. So last part of our webinar, I want to, all the speakers and the panelists to share your wisdom on the, future or the most pressing challenges of the immunotherapy or the immunodelivery field nowadays. And, as a biomedical engineer, pharmaceutical scientist, drug delivery people, how do we contribute our expertise to advance these fields? So let's start with our first speaker, Dr. Zhao.
Yeah, so yeah, it's a really kind of emerging area about immunotherapy and also like how we can use the drug delivery or like therapeutics to modulate our immune system. Because I feel like almost like every disease is related to our immune system. Not only the infectious disease, not only cancer, but many other diseases that is related. So one of the big challenges I feel like in this field is the kind of individual variability. So like when we see the outcomes of the immunotherapies, we always see responders versus non-responders, not only from the clinical trials, but also in our preclinical studies, we also see such bigger variations in many of our models too. So one of the things that we need to really think about is what kind of contribute to these variabilities in our studies. And like how we can address such issue. So, for example, that when we see the mouse models, from my perspective, we kind of really find that the mice with different microbiome, for example, have a big contribution to that. So sometimes when you look at the differences in the therapeutics, you might want to look at whether or not that could be an issue if you want to have a little bit of control, for example, for the mice to kind of eliminate that factors, or using some other ways to kind of better control the study. Yeah, from the drug delivery perspective, as people working in the drug delivery field, we also wanted to really address the kind of personalized medicine that we can also design better drug delivery tools to kind of minimize such variabilities by really looking at what caused the diseases in the patient and how we can address that issue.
Thank you so much. So our second speaker, Dr. Feng.
Yeah, thank you. Yeah, I think I totally agree with Shijie. Yeah, so personalized medicine is a key thing. Yeah, since for immunotherapy, especially, yeah, we have very kind of a heterogeneous response. Yeah, so I would like to think how to, I'm a device guy. I'm thinking how we can make a kind of a prediction using different kind of devices to help a clinician to really make a better companion diagnosis. Yeah, this is one thing. Then the second thing I also mentioned in my talk. So current cancer immunotherapy is pretty powerful for blood tumors. However, for solid tumor, it's still quite challenging. Yeah, I would also like to kind of explore or to really get everybody's attention to see how we can overcome the kind of or improve the therapy for the solid tumor immunotherapy. Yeah, that is some of my naive thoughts. Thank you.
Thank you so much. So, Zongmin.
Yeah, I think one pressing challenge is actually basically I think 4 words like where, what, how, and when. So basically, this that's actually 4 words. I think the pressing challenge for the immunodelivery, especially I say for where and what. So these are really pressing challenges because tumor let's say, no, not tumor. Immune environment is very complicated. And you have so many immune cells there, and immune environment is very complicated to figure out where and what to deliver. These are pressing challenges to answer. However, as a bioengineer in the field, probably we are not the best people to answer these questions. And this really comes from the better understanding of the immunology and the biology. However, but as a bioengineer, as a drug delivery people, what we can contribute is from how and when to deliver, right? So we can basically deliver technologies to figure out how to deliver them, how to target the specific immune cell populations, and also when control the release of these the payloads. So that's actually one of my perspective in particular in the immunodelivery field.
Thank you so much, Zongmin. Probably Guojun, so what's your opinion on this pressing change in the immunotherapy?
Yeah, so I fully agree all the comments or suggestions or ideas from the three speakers. And maybe while Zhongmin actually talked what I wanted to say, so maybe I'll add one more thing. So here is more from the area of cancer immunotherapy. So we know that this abscopal effect seems to be very valid in both preclinical models and also the clinical data or clinical evidence. So I don't know whether we could, in this field, I mean, we sort of acknowledge this effect, right? So, but how can we fully leverage this effect? Basically, right now, so the traditional like chemotherapy or other therapies are mostly using this IV injections to systemically deliver the payloads, right? But if we could fully leverage this abscopal effect instead of this systemic administration, can we, as a bioengineer or from a material design perspective, can we use this local thoughts or local idea, the administration approach to and then leverage this abscopal effect to fully like address some cytotoxicity, like the toxicity issues or the biocompatibility issues. So that's well, personally, I don't have the answer right now. I'm just bring this up to see if this can be done in the field. Thanks.
Thank you so much, Guojun. So maybe Sichen, do you have any perspective on this press challenger? We also want to get the perspective from the graduate trainee, so that we can, collect all these thoughts to promote the field moving forward. Yeah.
Yes, thank you, Dr. Hu. In my perspective for cancer immunotherapy, I'm still curious about the factors related to the cancer microenvironment, as it's a huge challenge for immunotherapy. And the extracellular matrix and many tumor-associated immune cells will have different functions in tumor progression, and it will also bring difficulties in the context of drug delivery. And I'm, as a PhD student, actually, I'm also curious, how can we actually push the real translation of this intelligent designs into clinical use, and how in the future, how can I learn to combine different strategies to enhance the immunotherapy outcomes? Thank you.
Thank you so much. So I think it's really thought-provoking discussions. And personally, I think, immunotherapy is certainly a very successful therapeutic approach in treating various disease. And, as the biomedical engineer, drug delivery people, so we, personally think we have a huge opportunity in contributing our expertise into advancing this field forward, especially, with the inspiration of the mRNA vaccine. So you can see how, excellent of this delivery carrier can contribute to the clinical translation and the commercialization of these emerging modalities of therapeutics. So again, in this, emerging field of immunotherapies, we still got a lot to do, but I think, still there's the huge, bright field in the forward. So again, thank you so much for all the speakers for the outstanding talks and the thought-provoking discussion from the panelists. So finally, we have a recognition for the speakers. It's a certificate. Hopefully can provide the recognition of your contribution to this fields. So first is for our first speaker, the first certificate for our speaker, Dr. Shijie Zhao from University of Washington, Seattle. So thank you again. And our second certificate goes to Dr. Feng from Indiana University, Bloomington. Thank you so much again for outstanding talk. And the final certificate goes to Dr. Zongmin Zhao from University of Illinois, Chicago. So thanks again for your outstanding talk. All right, I think that's pretty much for today's webinar. And for all the audience, our excellent talks continues. So we will have our 23rd I Can Use talk next Tuesday. The topic is about MEMS and the micro energy harvesting system. So I will see you next Tuesday. Yeah. Thanks so much for joining us today for the topic of immunodelivery. Have a good night.
不再是奇迹，不再是幻想，此刻真感觉全世界为我鼓掌。不必太在意身旁惊奇的目光，可以点点头，可以放声歌唱。我创造奇迹，我拥有梦想，我希望看见所有骄傲的脸庞。不再为曾经失败放弃或感伤，努力才是真的方向。I can， 没有什么可以阻挡心中无限的力量。